Hi! Today's video we are going to talk about experimental design and how to set up an experiment and all the different variables that go into it. I am sure you've probably talked about experimental design over and over and over again in all of your science classes, but it is really important because all science is built upon this design. So we're going to talk about it again, and we're going to go over it and over it and over it all year long. I will warn you, this video is a little bit longer because we do have a lot to go over, but it'll be worth it in the end. So the first thing I want to talk to you about is what the heck is experimental design? Well, it's looking at the scientific method. So science uses a series of steps called the scientific method, and we carry out an investigation. Okay, and that is just, the whole point of it is to organize your thoughts and search for knowledge. When you're talking about the scientific method, we're looking at 6 major steps. The first one is making an observation. Then you have to identify your question, create a hypothesis, actually design your experiment so you can test your hypothesis, collect your data, and then form your conclusion. So let's talk about the first one, observation. Observations are gathered through your senses, and it's things that you notice in your natural world. Okay, so an example of an observation, if you can see on the slide, says that maybe you notice many salamanders near a pond that have curved tails and not straight tails. That's an observation. You used your sense of sight and you noticed something that was a little odd. Okay, so once you observe something that you kind of have an interest in, you're going to create a question, or you're going to research that a little bit more. So we're going to take our salamander example and we're going to carry that a little further. So maybe a question might be, why do those salamanders near the pond have curved tails instead of straight tails? Who knows? Let's do a little research. We're going to set up an experiment that is based on that question. The third one is pretty important. It is creating a hypothesis. This is a suggested solution. A hypothesis is an estimated guess. You have a little bit of background knowledge, so you're going to make a postulate, or you're going to create an idea of why you think that might be happening. Okay, so here are some key things. It has to be testable. It needs to be written as an if-then statement. So if this happens, then this will occur. It's a prediction of an outcome. So if we're sticking with our salamander example, a hypothesis might be, if you notice salamanders having curved tails, then it might be due to pollutants that are in the soil. After you've created your hypothesis, the next step is to create an experiment. You need to make sure that it is in detail, and the whole purpose of the investigation is to test to make sure that your hypothesis is right. So when we test, we have to look at variables, and variables are factors in the experiment that are being tested. A good experiment is only going to have one tested variable, just one. You can test multiple variables through multiple experiments, but each experiment is only going to have one major variable. We'll come back to variables in just a second. The next thing you're going to do after you've collected all of your data is you're going to analyze it. You're going to look at the results of the experiment. So it could be quantitative, and that's looking at numbers. So you've collected data, you've counted so many salamanders that have curved tails. Or it could be qualitative, and qualitative comes from the word quality, and that's talking about the description of something. Your results have to be organized, and we put those into charts and tables and graphs, and we'll spend some more time in another video talking about graphs and how to put those together. But for now, you just need to understand that when you take all of your results, you have to compile them in some organized fashion, okay? So if we say there were 20 curly-tailed salamanders and 13 straight-tailed salamanders in area number 1, that is quantitative data. Quantity, we're looking at numbers. After we have all of our data and we've compiled it into some sort of understandable, organized fashion, a graph, we're going to create a conclusion. The conclusion is looking at the answer to the hypothesis based on the observations that you made during your experiment. So basically, did the data prove or disprove the experiment? Did it prove or disprove your hypothesis? So if we're talking about our salamander example, did the pollution cause the straight-tailed salamanders to be curly-tailed? In conclusion, my data showed that ... So now that we've talked about the different steps, which I'm sure you've gone over before in your other science classes, now we're going to look at how we design a controlled experiment. And this is going to be so important. We're going to talk about this all year long, so you really need to nail it now. It's important for experiments to have specific controlled procedures. All of the different factors of the experiment are called variables, and there's a few different kinds. The most important ones are the independent variable, the dependent variable, and the constants, okay? The independent variable is the variable that we're changing or manipulating, and I always remember it because if you go like this with your hand, it makes a little I. It's what I vary, okay? So the independent variable is what I'm changing or manipulating in my experiment. The dependent variable is the variable that is observed or measured during or at the end of the experiment. So I always remember it. So if this is independent variable, the dependent variable is what you measure, okay? And then the other one that I mentioned were constants. Constants are the variables that you could change, but we're not going to, because remember I said you can only change one variable at a time. You can only manipulate one thing in your experiment. So the constants are the things that we could manipulate, but we're not going to, because we want to make sure that our data that we get at the very end of it is because of that one thing that we changed. So a controlled experiment is working with one variable at a time, so all the other ones should be unchanged or controlled. If several variables are changed at the same time, then you wouldn't know which variable is responsible for the observed results that you came out with. An experiment is based on the comparison between a control group and an experimental group, okay? A control group is the one that you use in the experiment to compare with. We call that maybe the boring group, okay? They don't get any special treatment. The experimental group is the group that receives that treatment or the changed variable. These two groups are identical except for that one thing that you're manipulating or changing, that one independent variable. The control group is going to be the comparison, so it's the exact same as the experimental group, but that one change is being removed, okay? The experimental group is going to show the effect, hopefully, of the variable that's being tested. Fill these in your notes, okay? The factor that is changed or manipulated before you begin the experiment is known as the independent variable. Good. The group that gets the special treatment is the what group? Experimental group. Good. The factor that is measured or observed, so this occurs during or at the end of the experiment, is called the group that doesn't get special treatment or used as a comparison is known as and all of the variables that you could change but don't, so that all remain constant, are called So we are going to go through an experiment and we're going to identify the variables just for practice. And we'll practice more in class, but I want you to make sure that you get this in your notes. So let's suppose that you want to figure out the fastest route to walk home from school. You're going to try several different routes and time how long it takes you to get home every time. Since you're only interested in finding a route that's fastest for you, you are going to do the walking yourself. On one route, you take the route home that is the state certified route, which is determined the fastest. So when we vary the route, which of these options would you consider that? Hopefully you said it is the independent variable. It's what we're manipulating or changing. All right, the time it takes to walk home is hopefully you guessed, the dependent variable. All right, the next one. Keeping the same walker, which is you, leaving at the same time of day, having the same amount of books in your book bag, and walking on a sunny day, so we make sure that the weather conditions are all the same. All of those are going to be the wet. Hopefully you said the controlled or constant variables. You taking the state certified route, what the state says is the best route available, is what? That is the control group. And all of the other routes that you take are considered experimental groups. I hope you got all of those right. If you didn't, that's fine. We're going to practice some more. But if you didn't get them right, I want you to go back and look at the definitions of these so that you understand why those are the best answers. And make sure if you didn't get them right, that you write down some good questions in your notebook so we can talk about these in class. Okay, a couple more things. After your conclusion, you are not finished with your experiment. You should do this experiment over and over and over and over and over again, because we want to make sure that your results are accurate, and we want to make sure that those results come out every single time. Because if that happens, you can support your hypothesis. Okay? So in order to verify the results, your experiment has to be retested over and over again. The other thing I wanted to talk to you about is the difference between a theory and a law. A theory is an explanation for a broad range of information based on lots of evidence that support the hypothesis or the theory. It has lots of experimental proof. It can be applied to unrelated facts and new relationships that come about. It's flexible enough that you can modify it if new data or proof is introduced. Theories can't be proven. They can only be disproven. So examples of theory are the cell theory, the germ theory, and the theory of evolution. We weren't there when evolution occurred of the dinosaurs, but we have lots of proof, and that proof is called fossils, that support the theory of evolution, okay? The other thing is a law. A law stands the test of time, most often without challenge. It's experimentally proven over and over again, exponentially more than the data that supports a theory. It can create true predictions for different situations. It has uniformity, and it is universal. So a couple of examples are the first law of thermodynamics, Newton's laws of motion, and Newton's laws of gravitation. Here's the deal. Most of the stuff that we talk about in biology are theories. It's stuff that has so much evidence that supports it, but for whatever reason, it's not a law. Most of the laws come with physics, okay? Just a little FYI. All right, so here's a little review. I want you to pause the video, jot these questions down in your notebooks, and then I want you to answer them, and when we come to class next time, we will talk about the answers and go over them, and we will practice a lot more. As always, if you have any questions, make sure you write them down in your notebook and ask me either in class, or if we don't get a chance in class, you can ask me outside of class, and I will be happy to answer questions for you.